Hi everyone, welcome back. So this is your biology educator Divya Singh, and today I'm going to discuss very basic information about DNA. We all know DNA kya hota hai. This is very important. Okay, so we know what is DNA. This is genetic information contained in our body, which stores all the genetic information and passes on to further generation. Okay, so if we define it technically, so this is a long polymer of polynucleotide chain. So we'll learn what is polynucleotide chain, what are its components. But for now, let's learn the double helical structure of DNA. Okay, so how they were discovered, what all information did we get after their discovery. So this double helical structure of DNA was discovered by James Watson and Francis Crick. So these two were the responsible person for discovery of double helical of structure of DNA. Okay, but there is a very interesting information behind this discovery. How they came to this point, that was derived from data of X-ray diffraction pattern experiment conducted by Rosalind Franklin and Maurice Wilkins. So that led to derivation of this double helical structure of DNA by James Watson and Francis Crick. So let's learn about the double helical structure of DNA, what all things are there. So this is the sugar and phosphate backbone. This skeleton that you can see outside. Okay, this one. So this is sugar and phosphate backbone. I'll let you know what all these things are. But for now, I'm just labeling and letting you know the information. So this is sugar and phosphate backbone. Then this inner projection which you can see, this is base pairing. Okay. So this is base pairing of nitrogenous bases. And this is complete one turn of DNA. Do you know human DNA is of which form? This is one turn of DNA which consists of 10 base pair. 10 base pair. And it is around 34 angstrom in length. Okay. So human DNA is of B form. Okay. There are several forms of DNA, among which human DNA is of B form. And distance between two consecutive nitrogenous bases, as you can see this one, is 3.4 angstrom. Okay, so now can we calculate the length of an human DNA? See how this is calculated. Length of human DNA. So this is number of nucleotides into distance between them. So in a diploid cell of human body, there are 6.6 into 10 to the power 9 base pair, which is multiplied by the distance between two consecutive base pair, that is 3.4 angstrom, which can also be written as 0.34 nanometer. So this comes approximately 2.2 meter. So this is the length of a human DNA, 2.2 meters. Now, as I said, DNA is a long polymer of polynucleotide chain. So what are its components? So they are basically made up of three components. The first one is nitrogenous bases. You can match it with the diagram here. Now I'm writing it. So this is nitrogenous bases. Then what we have? We have a ribose sugar. Ribose sugar, which is a 5-carbon ring structure. That's why this is also called pentose sugar. Then we have phosphate group. So these three are the main components which are forming the polynucleotide chain. And now in this nitrogenous bases, we further have two types of them, which is purine and pyrimidine. So both are heterocyclic ring structure. Okay, but purine is double ring structure, this is single ring structure. And in purine we further have adenine and guanine. And in pyrimidine, we have cytosine and thymine. Okay, so this is about DNA. But in RNA, we also have cytosine, thymine, and uracil. So this is all about nitrogenous bases: purine and pyrimidine, adenine, guanine, cytosine, and thymine. So there is base pairing among them, which is depicted here, and their base pairing follows a rule called Ervin-Chargaff rule. So we'll discuss that rule in next video. So for now, that's all. Keep studying. Thank you.